Hello and welcome to this Visions 2020 panel, participating in natural history studies and clinical trials. My name is Todd Durham. I'm the Vice President of Clinical and Outcomes Research at the Foundation Fighting Blindness. You may have read on our website and press releases that we are conducting natural history studies through our consortium to generate data that can be used to design clinical trials. I expect many of you might be wondering what this is all about. What is a natural history study? How is it useful for designing clinical trials? And if I were to participate in a natural history study, what should I expect? And what is this consortium all about? To help answer these questions, I am joined by a panel of 3 experts who I have the pleasure of working with on these studies. Dr. Jacque Duncan is Professor of Clinical Ophthalmology at the University of California, San Francisco, and she is the Chair of the Foundation's Scientific Advisory Board and Study Chair of the Rush 2A Natural History Study. Welcome, Jacque. Dr. Rick Ferris was formerly with the National Eye Institute and is currently an independent consultant and vice chair of the Foundation's Scientific Advisory Board. Based on his experience with a similar collaborative research model, Dr. Ferris was instrumental in the establishment of the Foundation's consortium that conducts our clinical studies. Thanks for joining us, Rick. And Dr. Mark Pennesi is Associate Professor of Ophthalmology at the School of Medicine at Oregon Health and Science University and Study Chair of the Proice Natural History Study. And great to see you too, Mark. Well, let me start with you, Jacque. Here's a question I get frequently from colleagues here at the Foundation. What is a natural history study? Can you tell us a little bit about that?
Well, thanks for having me, Todd. It's a real pleasure to speak with the Visions 2020 attendees virtually this year. A natural history study is, by its very nature, an observational study that helps us learn more about how vision changes in patients who have retinal degenerations over time. It, by definition, does not include any treatment. So there's no treatments that are being investigated in a natural history study. But in order to know whether or not a treatment is helpful, it is critical to understand what would have happened in the absence of any treatment. And that's the information that we can gain from a natural history study. Natural history studies are very important for identifying patients who can participate in clinical trials and determining which measures are going to be the most sensitive and informative to study in response to different treatments. We can then compare the response to a treatment to the natural history of disease if there were no treatment to determine whether or not the treatment helped. So natural history studies offer an opportunity to learn more about how different diseases cause vision loss over time and how best to monitor them so that when treatment trials come along, we can be most likely to determine whether the treatment is safe and effective.
Great. And Mark, maybe you can expand on that. The FDA has a guidance document that stresses the importance of natural history data when planning drug development, especially for rare diseases. I wonder if you can tell us a little bit about how natural history data are used to design a clinical trial, and in particular, why this is important for rare diseases.
Mm-hmm. Well, anytime we have a new drug, we want to make sure it's safe and effective. And one of the ways of doing that is to look at different measurements. And you might want to see if a particular measurement improves or if it slows down in the context of a disease. And in order to know if that happens, you need to know what the natural history of the disease is and how much change you might expect over a given period of time. And without that information, it makes it hard to design a clinical trial. So having that information informs us as to how long the clinical trial might need to be, or how many patients we might need to enroll in order to see that change. And these things are important because you can sometimes get what is called a placebo effect, where if you give somebody a therapy, they might actually improve even without the drug being effective. So we need to know what the magnitude of that is as well. So natural history studies are very important, and especially in the context of a rare disease, we may not know a lot about the disease, and we may not have information on the history of that disease. And when you think about some of the forms of inherited retinal dystrophies that we deal with, any one expert in the country may have a handful of those patients, which is not really enough to calculate these rates of progression. But if everyone comes together and pools all of those patients together, we can start to get that kind of information.
Great. So one obvious difference between a natural history study and a clinical trial is that there is no experimental therapy in a natural history study. Jackie and Mark, are there other ways that natural history studies differ from clinical trials?
Well, the main one is that there is no treatment involved. In other ways, really, there are many similarities. The equipment that is being used, the technicians that are doing the testing, the investigators that are doing the study all have to go through rigorous certification procedures to be sure that all the measurements are being collected exactly the same way at each of the sites and the same way that it would be done if it were for a treatment trial to determine if something was safe and effective. Certainly the risk of participating in a natural history study is arguably less than participation in a clinical trial might be if there were any side effects or untoward effects, safety concerns about the treatment being investigated. But in truth, I think many of the measures, much of the experience, minus the treatment, is very similar between a natural history study and a clinical trial.
Yeah, I agree. I think they run in a very similar manner. We record the data in a very similar way where we keep track of everything. They tend to be a little less stringent in that you may not have as many visits. For example, in a natural history study, you might only come in once a year, versus in a treatment trial, you may be coming in multiple times in a month, even after a surgical treatment. So they're a little bit more relaxed in that sense, that the timing is less rigorous than an actual treatment trial.
Great. Well, Rick, let me ask you to join this conversation on the panel. In all areas of research, you can answer your study questions using different kinds of study approaches. And it seems as if we wanted to know about rates of disease progression, we have a lot of options. So we could go to Dr. Duncan's site and look backwards in time at how her patients have progressed. And we could go do the same at Dr. Pennesi's site. And we could put all their data together and analyze that. And that's what we would call a retrospective natural history study. But what are some of the issues with that kind of study, and are there some ways that we can do better?
Well, Todd, first I'd like to apologize for my laryngeal polyp, and I hope my voice hangs in there for the rest of this. It's the same thing that Adele and Julie Andrews had, but I'm not quite the singer that they were. Anyway, as you say, the retrospective observational study seems like a natural way of doing this. It's easy and it seems inexpensive, but there are problems. And actually, there are big problems. If we think historically as to how we had information on how disease progressed in groups of patients, it was astute clinicians would put together their cases, maybe just in their mind as to how things progressed, and they would use that information to tell their patients what they might expect with similar disease. Then we said, well, maybe we need a more data-driven approach. And as you said, you could go to the clinical records and collect charts and go annotate them. And back in the dim time when I was first starting, we would gather piles of charts and annotate them. Even worse, we might have to go to microfilm, and you get seasick just going through that process. And what you found at the end of that was that you pretty much had a mess. People would drop out. You know, some would be followed for 6 months, some would be followed for 16 years. They come in at all sorts of irregular times. And we thought the obvious solution to this was going to be the electronic medical record. Now we're going to be able to get all this information, and it's just going to be wonderful. Well, actually, 2 decades ago at the National Eye Institute, we set up a electronic medical record which was designed for research. So it should be ideal for this process. And maybe 5 or 6 years ago, when I was clinical director there, we thought, well, let's look at our Stargardt's patients and see what the natural history of Stargardt's is. Well, that electronic medical record was terrific at identifying who had Stargardt's disease. So we were able to quickly get all the patients with Stargardt's disease, but, and I should have thought of this 20 years ago, but I didn't. We still had the same basic problems, that there were losses to follow-up, and how do you deal with that? All the tests weren't done on everybody, so there was a lot of missing data, and the tests that were done weren't always done in exactly the same way, so you weren't sure how to pool that data. If it was a refracted vision and then you had a non-refracted vision, how do you put those together? And then patients coming in irregularly, when you're trying to say, well, I'd like to know what proportion of patients lose vision in 1 year, 2 years, 5 years. Well, the patients didn't come in at 1 year, 2 years, and 5 years. They came in when they feel like coming in. Some came in annually, but others came in at 8 months or 16 months. And at the end of the day, the data were very difficult, if not almost impossible, to put together. And that experience led us at the National Eye Institute to say, wait a second, if we're going to try to get this kind of information so we can report on what happens in Stargardt's disease, we're going to need to set up a protocol. We're going to need to have patients come in yearly, and we're going to put some boundaries around what yearly means, like sometime between 10 and 14 months, or 11 and 13 months, so that we're gathering data on each patient at the same time and in the same way. And if you think about it, that's what Jackie and Mark just described as what we're doing in the consortium. We have a set protocol, and actually, when you think about a clinical trial, the control group in the clinical trial is basically a natural history study. The problem we have right now, particularly in rare retina diseases, is what outcome variables are we going to use? And because we don't have the good data that we need, it's hard to predict how many people are going to lose vision or lose visual field over time. And that's the kind of thing that we're going to get from these natural history studies. We're going to be able to estimate how many people are going to lose vision over time if untreated. And then based on those estimates, we can make some pretty good calculations of how many people do we need and how long do we need to follow them.
Wow, that's great. Jackie and Mark, do you have anything that comes to your mind as you listen to Rick?
Well, I would say the way I think about a retrospective trial versus a prospective trial is it's a little bit like cooking. A retrospective trial is sort of like you open the refrigerator and see what you have in there and what you can make. Whereas a prospective trial is actually making a list and going to the grocery store and planning the meal ahead of time.
That's a great analogy. Thank you for that, Mark.
Can we use that in the future?
Sure.
Jackie, anything else? Or?
Yeah, no, I just think that the wealth of experience that Dr. Ferris brings to the consortium and the wisdom that Mark has acquired over his many years have contributed to the ability for us to design really interesting, exciting, informative studies that I think are currently underway. And I think they're going to be a lot, they're going to make major contributions because they have been so carefully designed and they're being executed in a way that is really rigorous and going to give us useful information.
Well, that's a great segue, Jackie. I wanted to just acknowledge that you and your co-authors recently published your first paper from the Rush 2A study in the American Journal of Ophthalmology. And I wonder if you can tell us some of the things that you learned from that first paper and how they might be useful for companies who are designing clinical trials.
Well, on behalf of the consortium investigators and the executive committee, 2 members of which are represented on this panel, Dr. Pennesi and Dr. Ferris, yeah, it's a real pleasure to talk a little bit about the RUSH2A study and the first manuscript, which was recently released online. I'm not sure it's officially out yet, but it has been accepted and has begun to be disseminated. The RUSH2A study enrolled 127 people with mutations in a gene called USH2A. And USH2A is the most common cause of Usher syndrome type 2, which causes mild to moderate congenital hearing loss in association with retinal degeneration. But it also happens to be the most common cause of autosomal recessive retinitis pigmentosa without hearing loss in people who have normal hearing at birth but subsequently develop retinal degeneration and vision loss. And it's not entirely clearly understood why that same gene can sometimes cause Usher syndrome and sometimes cause retinitis pigmentosa. So this study enables us to understand or to investigate some of the reasons, some of the factors that are associated with the diagnosis either of Usher syndrome or retinitis pigmentosa. The first paper that just came out is reporting at baseline, our very first visit, on the main outcome measure of the study, which is visual field testing using a standardized approach, same equipment, same certified technicians at every location, and with a software protocol of analysis that was developed at the KCI Institute, where Mark is on faculty. We determined at baseline in a cross-sectional way that the things that are associated with worse visual fields are duration of disease. The longer you've had the disease, the worse your visual field is. And even if you control for the duration of disease, people with Usher syndrome tended to have worse side vision than people with retinitis pigmentosa, given the same amount of disease duration. So that gives us helpful information as we embark upon the longitudinal aspects of the study. We have that information at baseline and we have something to measure going forward. We also know how that, we have a bunch of other studies underway based on that initial visit that are in various degrees of, or various stages of preparation. Many are about ready to be submitted and some are still being analyzed to look at other measures that we have collected, including measures of retinal structure with OCT, optical coherence tomography, a special kind of picture of the retina inside view, and that Mark is going to be leading, where he's looking at visual fields in comparison to the structural measures. We have another paper where we're looking at the very central visual field using a technique called microperimetry and how that corresponds to retinal structure in our study population. We are working on a paper to analyze hearing and smell function in patients with USH2A mutations, given that some patients develop hearing loss at birth and other people do not. And the gene is expressed at a part of the cell called the cilia, which is a very important structure also for smell and hearing, other different sensory organs. And a very important paper that's very interesting and just likely to be submitted soon is looking at the genetics, the different genetic risk factors for developing either Usher syndrome or retinitis pigmentosa. And that's being led by Rob Huffnagel at the National Eye Institute, who is an outstanding geneticist. And he's found some really interesting associations whereby different types of mutations and the number of severe mutations, which make the gene not produced at all, called truncating mutations, makes you more likely to develop Usher syndrome, but other specific changes make you more likely to develop retinitis pigmentosa, and certain genetic mutations predict whether patients will have more severe or less severe vision loss at a given disease duration. So there's so much we're beginning to learn even just after the first visit, the baseline visit. I'm really excited to see how this study will progress over time. We have a 4-year study following 105 of the 127 people enrolled. The 105 were enrolled into what we call the primary cohort. They were people who had earlier disease because they had enough visual field for us to measure visual field change over 4 years. The other 22 patients were in the secondary cohort, and they were seen just at the beginning visit, but will not be seen every year because their vision is at a stage where it's harder to follow meaningful changes over time.
Thank you, Jackie. The RUSH2A study is really exciting, really is already the results we're getting out of that study. Thank you. Now I'd like to hear from Mark again. Mark, you're the study chair for the Proice Study. What can you tell us about the rationale for that study, what we hope to learn from it, and can you maybe spend a few minutes to compare it to the Rush 2A study that Jackie just told us about?
Yeah, well, I think first off, we're studying a gene called EYS, which is one of the top 5 genes in retinitis pigmentosa. And so it's actually a fairly common gene, but it is a gene for which we really don't know a lot about, partly because there aren't good animal models of the disease. And so it's an opportunity for us to learn more about this gene, which is actually pretty common. Now, the natural history study that we are embarking on is actually quite similar in many ways to Rush 2A. And that's a great thing, because this is one of the great things about the consortium, is that we don't have to reinvent the wheel every time we do a trial. We can do a trial and then take the things that worked well and reuse those. And in a similar way, we learn mistakes that we made in previous trials, and we can correct those later on. So we are actually using a very similar framework for the EYS study. I mean, there are a few things that are different, because EYS doesn't have hearing loss, so we're not studying hearing and smell as we were in the Rush 2A study. But we are looking at many of the same functional outcome measures with vision, such as visual fields and microperimetry and OCT. But we've refined them a little bit based on what we learned from the Rush 2A study.
Jackie and Mark, you've already told us a lot about the research and the scientific aspects of these 2 studies. Now, maybe we can talk about some of the practical aspects. If someone were interested in participating in a study like this, what could they expect in terms of the time commitment and the clinical assessments that they might undergo while they were participating? And here I have to note and remind everyone that the Rush 2A study is closed for enrollment. We're following up participants in that study, but the Proice study is still open as soon as we can resume our research activities. So maybe you can tell us a little bit from the study participants' perspective what they might expect.
Well, I think the study participant should expect first a screening visit where the investigator will contact them and say, there's a new study that we're enrolling patients for, and we think you might be a good candidate. And they will then offer the candidate or the patient a consent form, or an opportunity to look at a consent form, so they'll understand what is involved with a particular study. If the person thinks that it's something they want to do, they think they have the time to participate, then they'll come in for a baseline visit where they'll have an opportunity to go over the whole consent form and ask lots of questions, sign the consent form after they're sure they want to participate. And then there will be lots of examinations, including questionnaires where we ask about how their vision loss has impacted their life, and questionnaires about their history and their medical conditions, medications that they take, different kinds of measures of visual acuity, how many letters they can read on a chart, measures of side vision. Many times we learned from one of the lessons learned from the Rush 2A study is that we probably don't have to do that test so many times. We can probably get away with fewer repetitions and still have reasonable results. Measurements of the central vision, pictures of the eyes, whether that be with dim red light or slightly brighter light, to get different information about what the retina looks like. The studies that we are doing also include measurements of nighttime vision. So the trials that since these are often rod involving diseases, patients have to be in the dark for half an hour to 45 minutes before additional testing goes on. The first test they might take might ask them to push a button when they see a dim blue, red, or white light. And then subsequently in the Rush 2A study, we did an ERG contact lens test to measure how well the retina responded to light under different conditions. there are other additional ancillary studies that are performed at some sites, but not others, where they measure the very central vision or take special pictures with special machines that are only available at select sites. So all in all, the first baseline visit sometimes took 2 days. Sometimes it took more than 1 day to accomplish the whole thing. The annual visits for this natural history study are completed within 1 day. So it's a big commitment at the beginning, but then the time requirement is less as time goes on. But people should expect to come back every year within, as Dr. Ferris was saying, within a window of about a couple of months around the day that they were there a year earlier, so that we know the information is being collected at roughly 1 year intervals. In addition, sometimes before you're known to be eligible, to confirm that you're eligible for the study, some genetic testing studies will be required. It's not required of everybody. It depends on how much genetic testing you've already had before, but some people will need additional testing to confirm that they are eligible, which may require some saliva or some blood. And I'll turn it over to Mark to get his input on what is going to be required for the Proice study.
So it's a very similar study, and one of the primary requirements is that you have 2 mutations in the EYS gene. And one thing that's interesting about genetic testing is that it's not perfect. And I always give the analogy to my patients that genetic testing is a little bit like fishing, where sometimes you catch a fish and sometimes you don't. And just because you don't catch a fish, you can't conclude there's not a fish in the lake. And so many times we will get genetic testing and we'll find the causative mutation, but sometimes we don't. And sometimes we get what we call an inconclusive result, where maybe we find 1 copy of a mutation in a gene that would usually require 2 copies of a mutation to cause disease. And so that's sort of inconclusive. So one thing we are doing with the EYS study is, if you've had genetic testing in the past that was inconclusive, and maybe you found just 1 copy of the EYS mutation, we'll try to repeat that to see if we can find that second mutation and really confirm that that is the causative mutation. And, you know, I think for all of our studies going forward, genetic testing is becoming a really important component. And that's something that Foundation Fighting Blindness has been really great about, is supporting the My Retina Tracker program, which has allowed us to, you know, test the majority of our patients now for genetics. And that's been really helpful. And that's really vital for natural history studies too, because we want to get patients into the right natural history study.
Great, thank you. And we've alluded to a few times the fact that these studies that are being conducted by our consortium, which now includes more than 30 research sites around the world. Rick, I was hoping you could tell us a little bit more about the consortium. After all, you've been a huge champion for this for us, and we really appreciate that. I wonder if you could tell us what the consortium's about. What does it accomplish that we couldn't do any other way?
So a little background. When I first went to NIH 47 years ago, I was lucky enough to be part of the diabetic retinopathy study, which was the first multi-center randomized clinical trial in ophthalmology. And it set the stage for a whole series of clinical trials over the last 50 years. And the problem with the studies, the way we were doing them at NIH then, was that every time you had to get a new contract, a new grant, the study would be set up from scratch. You had to get a coordinating center, data center, reading centers, choose your clinics. You had to certify the clinics. It was a very inefficient process, and I don't think any got started within a year. And typically, it might take 2 or even more years to get a study started. Well, thinking about collaborating with industry, and you said, well, we'd like to work with you, and we'll be ready 2 years from now. you can guess how enthusiastic your partner would be. So we were fortunate 20 years ago or so when Congress gave $100 million focused on type 1 diabetes. And we put in an application for some of that money to set up the Diabetic Retinopathy Clinical Research Network. And part of that whole idea was to become much more able to move quickly and to do multiple studies. And if you think about it, it makes so much sense. We have one coordinating center. We have a group of reading centers for various image analyses, visual field analyses, for example. We have over 100 clinics, and clinics can come into the study or leave the study. So it's not the same group of clinics that we started with, although most of them that were there at the beginning are still part of that. And over these years, we've done over 24 studies. And the ideas for new studies can come from investigators. They can actually come from anybody who wants to put in an idea. And so this was a tremendous success for diabetic retinopathy research. Well, thinking about it from taking a step back, if it was useful for diabetes research, it's even more important for rare retina diseases where a single clinic might get hundreds of patients with diabetes. Mark's clinic probably doesn't have 100 EYS RP patients, and neither does Jackie's. But already the consortium has 30 clinics, and they're international. Each clinic has gone through a process of standardization and been certified already to do procedures, which, as Mark said, are going to be typically done across studies. The natural history studies I look at as the first steps. And one of the important first steps that we don't think about much, but has been very valuable in the natural history studies, is we have gotten these sites certified. We have gone through some of the difficult times of getting standardization so that when we're ready to do clinical trials, we're not going to be so worried about the data we're collecting and the connections with the coordinating center that have been set up. And we're now developing outcome variables that have been done in a standardized way and have predictable event rates so that when somebody wants to do a clinical trial in USH2A, we can say, well, here's what you're going to expect without treatment. And now we can help you calculate how long of a study and how big of a study we need to do. And then there's one other thing that you don't think about much for natural history studies, but I think is going to be true here. Think about the USH2A study. We're going to continue to follow these patients for years. And if we had a treatment that we wanted to start next year or 2 years from now, we've already got a cohort of patients that we've identified. We don't have to go out in the bushes and find them. We already have a group, and inevitably we're going to need more than that group. But it's very nice to get a head start in recruitment. So I think this consortium has put us in a great position to start doing clinical trials. And it's a pretty timely development, given that there's a lot in the pipeline right now in rare retina disease that wasn't there 20 years ago.
That is so true. We're really excited about that. I wanted to transition from sort of where we are today and ask each of you over the next 5 to 10 minutes to think about what's coming in the future, and starting with considering the fact that we have this consortium, this research infrastructure developed at the moment, and we're doing these 2 large natural history studies. Maybe I'll ask Rick first, and then Jackie and Mark each to respond to this question. But what, other than natural history studies, what are some things that are possible for us in the future? What do we have to look forward to?
Well, so I said these natural history studies are setting the stage for clinical trials, and that's what all of us are really interested in. How do we develop new treatments that change the natural course of these diseases for the better? And as I just said, the consortium seems to be the perfect platform for multiple clinical trials. We've got multiple rare retinal diseases, and we really need to collaborate to advance this field. And I'd say importantly, this is a great opportunity for government, academia, and industry to collaborate. And as an example, people might say, well, industry is going to be reluctant to collaborate. Well, with the DRCR, more than half of the funding has come from industry and collaborations with industry. So I think it's a tremendous opportunity. And rare retina diseases have particular industry issues where the collaboration may really be particularly important. So I think this is a great first step.
Yeah, I agree wholeheartedly with that. And I do think industry will start to partner with us. And when you think about it from their perspective, a phase 1/2 trial might cost $5 to $10 million, and a phase 3 trial can be $30 to $40 million, and they don't want their trials to fail. And one way of really helping that is to have good natural history data. It really can shore up and really serve as a foundation so that you don't miss an effect. One of the worst things would be to have a drug that works, but come to the conclusion that it doesn't because you didn't have the right natural history data. And so I think, increasingly we will see an investment in natural history studies and hopefully an investment in our consortium, for doing them, because I think we're really well poised to carry out those tasks.
I would echo what both my colleagues have just said. I also think, industry, one of the big expenses is how long the trial takes. And so if they don't have to wait around for us to get certified and have the right technicians and find the patients, it's going to save them money in the long run. I think an hidden gem, kind of unexpected benefit that the consortium is likely to bring, if it's at all anywhere near as successful as the DRCR.network was, is I think that, again, this is a really exciting time in the field of retinal degenerations, with so much more understood about mechanisms of disease, so many potential therapies on the horizon, so many trials getting underway. I think the consortium really is inspiring interest in the field in a way. It's increasing visibility and awareness among patients and among retinal specialists. I think there's more interest in this field than there has ever been traditionally. And I think the consortium, the DRCR has over 100 different centers. We have 30, but I think that's an increase in interest and expertise in retinal degenerations will be beneficial to patients whether or not they participate in a trial, because they are more likely to be able to find somebody with expertise in retinal degenerations to go see and take care of them. That also may be able to inform them about trial opportunities and the latest therapies as they become available. So I think there are so many benefits. I'm really thrilled that it's up and running, and I look forward to a really bright future ahead.
Wonderful. Well, I'd like to end this Visions 2020 panel by asking each of you to think back on the field when you started your ophthalmology career, whether that was in clinical care practice or in research, and think about where we are today and what excites you the most about where we are today and why. So maybe we'll start with Rick.
So when I started in ophthalmology almost a half century ago, there were no effective treatments for diabetic retinopathy or age-related macular degeneration, let alone rare retina disease. And over my career, I've been fortunate to be involved in a lot of clinical trials that have wound up with identifying very effective treatments. Reduction in vision loss in diabetic retinopathy is about 95%. compared to where it was 50 years ago. And AMD now has treatments. We still have some work to do with AMD, but we, like with rare retina disease, there are a lot of avenues that have developed recently that are exciting and can move things forward. But I look forward to doing the same kind of things that we've done in diabetic retinopathy and AMD, but in rare retina diseases. And as Jackie was just saying, fortunately, there's a lot in the pipeline. It's a really exciting time. And I think setting up this infrastructure is really exciting.
And how about you, Jackie?
I would just say, looking at the experience that Rick has just outlined over the last how many decades, makes it really, I feel like when I was a resident, I felt like there were a couple of unsolved fields in ophthalmology. There were a couple of problems that we didn't have good answers for. And retinal degenerations was like the top of the list. I felt like these are diseases that patients were born to have due to a change in their DNA, not through anything they did wrong or anything they could have done differently, that were beginning to be understood at a molecular level, beginning to be understood at a cellular level from animal models, and just beginning to be understood from clinical reports that were somewhat few and far between. And fast forward, 20 years, at the time when I was interested in this, it was largely because I was interested in the scientific perspective. It was so clean with the single gene causing the disease, unlike AMD where it's much murkier. I thought it was a really prime sort of model for developing therapies that might help patients. And now, I feel like I stumbled into something that makes me look a little prescient, although I think it wasn't quite so intentional. I feel like the luckiest person in the world because I ended up choosing a field that has limitless potential for helping patients, and so much progress is being made all the time that I just couldn't be more excited about the way the field's going, the developments that are taking place that are going to enable really great, promising developments in the future.
How about you, Mark?
Yeah, I mean, I echo Jackie's comments. I think what drew me to this field was learning that there were really no effective treatments at all for this disease. And, in the short time that I've been here, we've now have the first FDA approved gene therapy for a rare disease. And we're actually treating patients, with that and seeing like an effect and seeing how it changes people's lives. And that's pretty amazing. And I do feel very lucky in terms of the timing of that. But, also that's still a very small number of people that we have a treatment for. And there's so much more work to do. But on the other hand, we have 25 some more trials going on for different inherited retinal degenerations. And I think the field has finally gotten attention that it deserves from industry. And, it seems like every other week I'm talking to a company that has some kind of new innovative technology that might be able to be applied to inherited retinal dystrophies. And so that's just really exciting to have so many different potential treatments. And we know that they're not all going to work, but some of them are. So we now have to kind of do the hard work to figure that out.
Great, thank you. Well, this has been a lot of fun. I would like to thank our panelists, Dr. Jacque Duncan, Dr. Rick Ferris, and Dr. Mark Pennesi. It's a real honor and pleasure to work with you, and I know that our Visions 2020 attendees really appreciated hearing from you, so thank you so much. And thank you for your interest in Visions 2020.
Thank you.
Mm-hmm.